All right, hello everybody. Welcome to virtual San Diego Chromatin Club, our now online bi-weekly epigenetics seminar series. Well, we've been doing this for about a year now, over a year now, which is crazy because I don't think any of us thought we'd be doing this for this long. But we've gotten great feedback. We're having a good time doing it, and we're sharing some great science. It's been a blessing for everybody. I want to say up top that we've been very happy and excited to do this. But we will be taking a brief summer break for about a month. But we will be doing this all over again this summer with a whole new lineup of new speakers. So keep on the lookout. So the best place to keep up to date with the new speaker announcements and our summer schedule would be at our Center for Epigenomics website at epigenome.ucsd.edu. You can email us at the Center for Epigenomics and get on our mailing list by emailing epigenome@ucsd.edu. The best place or a good place to keep up to date are following us on Twitter, the Center for Epigenomics at CEpigenomics. Follow our organizers at Ani Deshpande Lab or at Hargreaves Lab. And also you can, of course, follow our industry sponsor, Active Motif, at Active Motif. And of course, a year in, I do want to thank all the organizers for this past year of wonderful science, including Drs. Bing Ren, Ani Deshpande, our host today, Diana Hargreaves, I guess I can thank me, and of course, Ms. Lauren Thomas, who really is the engine behind this whole seminar series. And with that, let me hand it over to our host today, Diana.
Great. Yeah, it's been super fun, and I'm thanks so much everyone who has participated over the last year. Today we're going to round out this first little segment by hearing from two of our rising stars right here in San Diego, Dr. Sascha Duttke and Owen Chapman. So Sascha comes to us from Chris Fenner's lab here at UC San Diego Division of Endocrinology. He's completing his postdoctoral training. Sascha received a Ph.D. from UCSD under Jim Katanaga and then performed a first postdoc with Chris Glass. He is interested in understanding the grammar of TF interactions with chromatin using bioinformatic and newly developed epigenomic techniques, including the recent technique he developed to measure nascent transcription called capped small Rna-Seq, which I think he'll talk to us about today. Sascha is a K99 awardee and currently on the job market. So take it away.
Sorry, couldn't find the unmute button. Sorry about that.
Okay.
Thank you very much for the kind introduction. I'd like to start off with the most important slide, which is basically acknowledging the people who did this work. Of course, Chris Fenner, an outstanding mentor. So if you're looking for a postdoc, definitely check out his lab. As a Benner lab is mostly bioinformatic, I really am grateful to have this talent that helps me with the analysis, with which Max Chang, Sam Roth, and also Nathan. The lab is performed in the lab of Sven Heinz, in the wet lab of Sven Heinz. Carlos did a lot of work, and we collaborate a lot with Aaron Collin from the Division of Infectious Diseases and his postdoc, Charlie Shi. And I'll also briefly touch on some COVID work that we did in collaboration with the ICU by Michael Lamb and Nicole Kufal. And throughout the talk, I will also try my best to point out the names of the people who did their work on the bottom of the screen. Of course, I have to thank our funding and also my wife for all her support to even being able to do science, and Active Motif for not only providing gear and clothing to us postdocs, but also for being the sponsor for the seminar throughout all these years, which was so much fun. Top right, there's a little screen for your Zoom window. If you want to use that, then you don't have to move it around a lot. So before we spend time on anything, we want to ask ourselves, why is it important? And the basis of nearly all biological phenomena, whether it's development, pluripotency, immunity, as well as diverse diseases, most cancers, autoimmunity, and so forth, can be traced back to proper or improper regulation of gene expression. At the same time, gene regulation, so changes in which genes are on and off and how strong they are, is what allows us to respond to life and interact with the environment. It is fundamental to understand eukaryotic gene regulation. And for this reason, it is of strong interest to understand the DNA binding motifs, or the DNA elements, the factors, as well as the mechanisms by which genes are regulated. As such, the gene regulatory mechanism is sometimes also referred to as the control center or the brain of the cell. Well, our DNA is the template of gene regulation. And one fun fact that potentially surprised you is that DNA has amazing storage capacity. And as published in the science paper here, 1 gram of DNA has 250 million gigabytes potential storage capacity, which is 250 petabytes. But so actually all the data in the world could be stored in a 2 by 2 by 2 cubicle if we would save our data in DNA rather than in hard drives. But there's a little other fact, is that our genome is only 6.41 picograms. And if you do the math, that's basically 2 times 750 megabytes. So another way to present gene regulation is the mystery, how to create a cute little being like that, or yourself, from 2 blank CDs. So how is that done? Well, one fascinating fact is that the DNA has several codes. One of those codes is quite well understood thanks to, I think, really elegant experiments by Nierenberg in the 1960s, who basically defined poly-U as the first word in the chemical dictionary of life, right? Poly-U encodes for phenylalanine. And due to follow-up experiments, we now have a solid understanding of this code of all these genes, or the things that make things happen in the cell. And by now we are familiar with why and how mutations in genes, whether it's in plants or whichever other organism, can have a tremendous impact. However, if we look at it, genes actually only correspond to about 6.7 or about 7% of the GWAS hits. Or in other words, the vast majority of mutations associated with disease are non-coding. Whether it's SNPs, but also the evolution of enzyme activity, or here the fun wing patterns in butterflies, the majority of the evolution is non-coding. So the DNA has 2 codes in one, and the second code is the non-coding or regulatory DNA. This is the code who instructs how the coding DNA or the genes are interpreted. And this code is still at large enigmatic. So it's important to ask, why has it been so hard to decode regulatory DNA? Well, of course, there are many overlapping codes and programs. So in you and I, there are thousands of transcribed loci that are active, and most of them have a unique program. Unlike DNA, regulatory regions evolve rapidly. So for example, comparing humans to mice, only 50% of the promoters are conserved, 50% completely relocated. And of course, regulatory networks are highly convoluted. This is because you have one DNA, but hundreds of different cell types. So in each regulatory region, only a fraction of the DNA motifs are active, but which ones? And further confounding the fact, of course, is that there's still a fundamental lack of knowledge. So the binding site for 40% of human and the vast majority of eukaryotic transcription factors is simply unknown. Consequently, we do not understand how transcription from 75% of human genes, and obviously most transcribed regulatory elements, is regulated. One brief example for the interdependence of transcription and translation is here shown. So basically, when we compare these 2 cell lines, the MCF7 cell lines predominantly starts at this nucleotide here as the A, while HeLa starts here with this TCT motif. And as you can see, this is a short truncation of the UTR. But now, when we treat those cells with mTOR or with taurine, which is a potent inhibitor of the mTORC1 complex, and the mTOR complex is one of those complexes that regulates cell growth in response to growth factors and nutrients, you can see that in HeLa cells, RPS11 translation and therefore the protein is repressed, while in MCF7, this repression is evaded. So as mTOR controls cell proliferation in response to these factors, now you can imagine that in one cell type, the cells keep on growing, while the others are repressed as it should be. So this change in the transcription start site circumvents mTOR-dependent translation regulation, and thus uncontrolled cell proliferation eventually leads to cancer. While the details here are really not important, I think this is a great example how and why accurate transcription start sites are meaningful, both to understand biology and the mechanisms behind it. As such, the transcription start site is sometimes referred to as the heart of gene regulations, where all the transcription programs and signaling is integrated to respond to the cell or the environment. And if that is too simplified, you can pick whichever busy slide or your favorite schematics, and you will see that all the signals eventually converge to transcription factors, which go into the nucleus, cause some change in response. When we talk about gene regulation, it is important to mention that we at large talk about RNA polymerase II. Polymerase II transcripts, however, in a tiny minority. Whether by mass, here the mRNA long non-coding ones, or by molecule number, we see that they're in a minority. Unfortunately, there is a magic mark that allows us to easily distinguish RNA polymerase II from other transcripts, which is the cap. So Pol2, but not Pol1 and Pol3 transcripts, get this cap added about 18 to 25 nucleotides into transcription. So this allows us to distinguish RNA polymerase II from other transcripts and will become important later in the talk. So to just briefly summarize this introduction is that the basis of nearly all biological phenomena can be traced back to proper or improper expression of genes, and Pol2 transcripts are in a minority, but capped. So when we talk about gene regulation, it is important to note that it is not the same as capturing the collection of stable RNAs found in the cytoplasm, which we commonly refer to as steady-state transcripts. These transcripts are captured by methods such as Rna-Seq. The presence of RNA in the cytoplasm is dependent on their stability. And indeed, the majority of Pol2 transcripts, when they are transcribed, are actually unstable. The presence of a gene or a transcript in the cytoplasm does not necessarily mean the gene is currently actively transcribed. On the other hand, capturing ongoing or nascent transcription allows you to generate a snapshot of which genes and transcribed regulatory regions are on or active at a very given moment in time. This is not only important to capture transcripts independent of their stability, but also to capture the complete transcriptome. So an example here, let's look at this a little further. This is one of our favorite loci, the SPE1 gene in mice, which encodes PU1. And when we do RNA-seq, we see, okay, these are the expressed exons. But now, if we add some other methods like cs-RNA-seq and growth-seq, which we'll talk about in a minute, you will see there's a large plethora of transcripts that become visible. The main difference between these methods is that cs-RNA-seq only captures the start site, while growth-seq captures the whole transcribed area. But note that these plethora of unstable transcripts also correlate with other hallmarks of active transcription, such as open chromatin, K27 acetyl, or K4 dimethyl. So a brief summary here. Basically in bold, you see genes and some non-coding RNAs that might be captured because they are stable by Rna-Seq. But using nascent methods, we get all the enhancer RNA, divergent transcripts, intronic eRNAs, antisense transcripts, the pre-microarrays, basically the whole plethora of transcripts. Why should we care about that? Well, as illustrated here, the most binding sites for lineage-specific factors, such as PU1 here, are actually in distal regions. And these are also the regions where, in case of PU1, that is required for both B cells and macrophages, the differential binding is also distal. So if you want to understand gene regulation, whether it's signaling or lineage determining factors, it is important to capture the complete transcriptome, including the distal regulatory elements. Furthermore, nascent transcripts are much more sensitive to capture changes at an order of magnitude more than steady-state Rna-Seq. And this makes sense because you only capture one moment and not sort of the whole baggage that the cell had for weeks and weeks while it was in culture. As shown here, we took mouse macrophages and we treated them with a KLA, which mimics a bacterial infection. And as shown, about 360 genes or so are differentially expressed using Rna-Seq, while you're in the 20,000s using cs-RNA-Seq. So it's needless to say that these methods are much more powerful for identifying the factors mediating changes in transcription So now you might ask yourself, well, why don't everybody always do nascent methods? And this is because previous methods, which predominantly included Pro-seq and Grow-seq, and were pioneered by Core, Waterfall, and other people of the Liz lab, they're actually quite difficult and laborious. Let's quickly go through that. So briefly, first of all, you need to make nuclei. This is quite easy if you have cell lines in the dish, but once you have plants that have secondary cell walls or metabolisms, it becomes more challenging. Well, then you run on the nuclei with an artificial nucleotide, which is either UTP for Grow-seq or biotinylated nucleotides for Pro-seq. Then you IP them 3 times before you do the library preparation. Another fact that complicates this method is actually that cells or tissues, each cell type is differentially sensitive to both shearing forces and detergent. For example, when you would use a liver, the difference between the hepatocytes and the macrophages in the liver, which are the Kupffer cells, is 100-fold. So if you use detergent lysis, you inevitably deplete some cell types, in this case the Kupffer cells, while keeping others alive. But the main point is really, this is laborious. I think I could do about 40 a week if I'm working hard. And so the method is largely limited, as I said, to cell lines and to small numbers. And to overcome this method, we developed Rna-Seq. And it's actually strikingly simple. And I guess we were surprised ourselves how well it worked. And the idea is, well, if you look at all the stable RNA in a cell, there's a minimum size, which in humans is usually around 62 nucleotides. If we now look at all the transcripts that are kept and smaller than these 62 nucleotides, what do we get? In addition, we generate a short input RNA, which is basically just all the short RNAs independent of the 5' marks, which serve as an input. And this little niche is only required if you have bad RINs or degradation, because that helps you to also work with RNAs that are a bit more degraded. If you have perfect RNA RNA, you do not need this. So briefly to go through the advantages and disadvantages, and the method is published now. Of course, the crazy advantage of this method is that we can use total RNA as input, which not only makes it easy for collaborations, but it opens up nascent transcriptomics to basically any sample, fresh, frozen, banked. You can look at your fixed cells after you sorted them, and even samples that have cell walls or whatever, you can use this method on. So it works with most eukaryotic organisms. It's a robust and relatively easy assay. And by easy, I say I did over 400 a week when we had to during COVID. It's very sensitive to regulatory changes, as shown, and it gives you single nucleotide resolution. And this will make more sense towards the end of the talk, but we propose Rna-Seq as a replacement for H3K27 or settle. And as you see here, the method accurately captures enhancer RNAs at single nucleotide resolution, similar to Pro-seq or GrowCap. But as this is an enhancer RNA, these are not captured by steady-state RNA sequencing methods. There are also some disadvantages. The transcripts are short and therefore inherently not allele-specific. At the beginning, you might struggle with carryover or small RNAs if you don't do the cutting right. The method will likely never be single-cell RNA-seq. And it is because you need about 1 microgram of input, although we were able to scale down up to 100 nanograms recently. So in summary, cs-RNA-seq is a very simple method to capture the transcription start sites of active transcription from any tissue where you can actually isolate RNA from. So needless to say, this methodological advance opened up numerous ways to study gene regulation. And for the sake of time, today I'm not going to talk about how this method allowed us minute time courses of signaling responses at minute resolution that really resolved the whole pattern of NF-kappa B activation in macrophages. I'm also not going to talk about, which is sort of my K99 startup help, how it opened up nascent transcriptomic to virtually all eukaryote and provided insights in the evolution of transcription factors. And unfortunately, neither, and you might want to invite Michael Lam, are going to talk about how cs-RNA-seq helped us to reveal diagnostic signatures from COVID ICU patients. So basically during clinical trials, blood was drawn. And as you can just briefly see here in this simplified UMAP, from the blood, we get about 300,000 transcribed regulatory regions that we can then, that are spread across different cell types. And then we can correlate them to those patients that recovered fast and left the hospital and those who had a very unfortunate outcome. And we can actually see that certain signatures like the STAT motif in neutrophils was a signature from a sad outcome. What I really want to spend the next 10 or so minutes on is to briefly talk about you how this method helps us to understand how transcription factors interpret our genome. The key point I want to make is that a transcription start site is a hallmark of gene regulation. And as mentioned, small Rna-Seq captures this at single nucleotide resolution. And this is a critical difference to methods such as ChIP or ATAC-seq, where you just get a broad peak. And when we now map the position of motifs relative to transcription start site, as you see here, we see distinct preferences that are conserved across cell lines, even species and methods. For example, the Tata box is at -30, at -50 we see SP1, and YY1 is more at +10. This spatial relationship would not be able to be resolved if you use ChIP-seq. But we were also wondering, this is the basal transcription machinery or the core promoter where there sits. So transcription factor binding here should be mutually exclusive with RNA polymerase transcription. And to gain insights if there might be potential differences of what the factors do based on where they bind, we did a simple experiment and just decided to knock down NF1 and YY1. And the results were quite striking to us. So if we sort our results by those TSRs or regulatory regions that were activated versus most repressed, we find a striking bimodal distribution, meaning that all the regulatory elements that had an NF1 site upstream of the start site appear to be repressed, while those ones that had an NF1 site downstream were upregulated. Well, for some transcription factors like YY1, which is yin-yang, it is known that transcription factors can activate or repress, but NF1 is known as a strong activator. And this striking finding to us that transcription factors can repress or activate transcription based on their relative position to the transcription start site was actually found around numerous factors. It's a little bit harder to see here in YY1, but YY1 has this sharp preferential peak pretty close to the start site. And as you can see also here, we see enhancing of the factors that have YY1 really close to the start site, while an induction or a de-repression upon knockdown when it's further away from the preferential spacing. So this proposes a model by which polymerase II is recruited based or repelled based on the innate biochemical properties of the sequence-specific transcription factors. So how can we test this model? And before we go through that, I wanted to introduce another quick methodological innovation. And this is GrowClick. We briefly talked through GrowSeq. GrowClick is pretty much the same, just that we use EUTP to run on the RNA and then click on a biotin. This advantage is because if the nucleotide is already biotinylated, the polymerase stalls, which makes it less sensitive. But biotin is the best mark to IP currently. And these, let's say, improvements allowed us to now do GrowSeq from as little as 100,000 nucleotides, as shown here, for naturally capturing the enhancer RNAs of this microloci. So cGrow is remarkably sensitive. And this actually allows us to now also capture transcription start sites from the genome, but also from plasmids. So as shown here for the SF3BP promoter that was kindly provided by Carlos Guzman, what I see is Rna-Seq over GrowSeq, these are the patterns in the genome, and this is the transcription start site pattern in the plasmid. Why is this a major advance? Well, there's little doubt that massively parallel reporter assays, or MPRAs, to not twist my tongue too often, such as StarSeq or SURE, have revolutionized our understanding of gene regulation. However, one limitation that we had so far is that we didn't have methods sensitive enough to capture the start sites. Hence, we use proxies such as reporter genes or resistances. But with the start sites, and what I've shown you before, we can now really decode the grammar of gene regulation. So with 5' cGrow and MPRAs, we can now assess how regulatory DNA, the blueprint, recruits different types of transcription factors to effectuate transcription. And as a quick reminder, plasmids can be super easily manipulated. We can clone oligos that have stretches of NNNNN. We can use E-blocks or synthesized oligos pools. I mean, the world is yours. And so as a proof of principle, The initiator is sort of a motif that accommodates the transcription start site. And we just took these 7 nucleotides and added N and N and N. We added a barcode, as transcription commonly starts in the middle of this motif. We transfected these millions of plasmid in cells, sequenced the plasmids, and did 5' seq-row that gave us the start sites and the barcode, calculated enrichment, and boom, here we see our initiator motif. Needless to say, this is just a proof of principle. There's lots of fun things that we can do with this technology. And one was, okay, let's test our methods. To do so, we said, okay, what happens if we take the YY1 motif that is preferentially usually at +10 and shift it 2 base pairs everywhere from -100 +40? Yes, that's 70 constructs, but remember, we get about 3,000 oligos for about $2,000. So we can do relatively 40 of such screens with one single library. And as a quick reminder, the YY1 motif is preferentially at +15, and as shown here, the transcription activity of YY1, it is largely inactive till it hits its preferential space around this site. So YY1 motif position affects the frequency of transcription initiation. And strikingly, this data are also similar to the pattern of what we've seen in the knockdown. This opens up an even bigger question, which is which transcription factor motifs out of the so many hundred identified functionally impact RNA polymerase recruitment, and where? And to do though, we used some data and the HOMA motif library and decided a simple protocol. We took a plasmid and designated 5 different spots, and then cloned this motif in one out of these 5 different spots. So to our surprise, the first thing is, and I want to keep it simple here, that there were only a few transcription factors that actually strongly activated transcription. 4 of them are shown here, but you can see again that GFX, NF1, and SP1, who preferentially bind at -50, had their strongest activation exactly in that region. -20 to +30, quick reminder, is where the base of transcription machinery binds. Binding of this factor was at large repressive to transcription, similar as shown in the knockdown data, with exception of YY1, which, yes, likes to bind at +10. So as a quick summary, methods play a central role in advancing science, and I briefly talked here about CS-Rna-Seq that is published and 5' seq-row that we hope to publish soon. And it really is fun because it breaks the barrier of what was previously known. And we propose that most sequence-specific transcription factors can activate or repress transcription based on their relative position to one another and the transcription start site. And a little point for those who are really into transcription, you can see that Tata boxes who really anchor down TF2D have a special rule, but even that we were able to decode using that. In other words, sequence-specific transcription factors guide transcription start site selection based on their biochemical properties. And that spacing is a key parameter to decode regulatory DNA. So again, these were the wonderful people who did that work. And as a take-home message, understanding gene regulation is critical to nearly all biological phenomena. I think that's nothing new. These are the methods that helped us to advance at least a part of the field, we believe. And understanding how and which sequence-specific transcription factors mediate the start of transcription is fundamental to understand gene regulation, also dispersed transcription, and mainly how our non-coding DNA works, how our non-coding DNA regulates the coding DNA. Or the mystery, how 2 blanks, CDs, are able to make a marvelous being like this human. Yeah, as mentioned by Diana, I'm on the job market, so if there's any interest, this is my email address. I think you need to be a little bit quicker because decisions will be made soon, but I would be excited. And with that, I thank you so much and give back to Diana.
Great, thank you. That was excellent. We do have a couple questions. Question from D. Gilbert. How do you distinguish a TSS from an enhancer RNA?
Oh, we just call them TSRs. We can distinguish them because Rna-Seq captures only stable transcripts and seq-row captures them all, or CS-Rna-Seq. And so basically those that are strongly expressed and only found in CS-Rna-Seq, we would call unstable, and those ones who are in Rna-Seq we could call stable. I personally avoid the word enhancer because to me, enhancer implies functionality. That's because I come from the flight Drosophila field. And I think in humans, enhancer is basically an unstable RNA and you don't have to show functionality. So that's why I try to say stable and unstable transcripts, but I think most people would call the unstable ones just enhancer in the current jargon. Yeah. Good question.
Yujiro says, thanks for the great talk. Could you please comment on how Rna-Seq compares to cage-based nascent RNA profiling methods such as NetCage, which are claimed to perform similar tasks?
Yes.
Yeah.
So NetCage is basically well, it's tricky to say that in someone. What they basically do is they isolate chromatin and then do cage on the chromatin-associated DNA. The first problem that we experience with NetCage, except from the fact that it's not quite as easy as Rna-Seq, is if any RNA is stuck to the chromatin, it is defined as nascent. The second thing is you have template switch, which depends on a CG change. And at least in my hands, template switch protocols, because the RT adds an extra G on the side, and then you have a CG ligation. A lot of transcripts actually start with a C, and those that start with a C are highly upregulated in NetCage towards Rna-Seq. So I can't fully comment on it because you need to be an expert in NetCage to do the methods. But in our hands, Rna-Seq is, I mean, it's basically like Rna-Seq with a gel. It's super easy. While in NetCage, I don't claim to make anything because you need to be really good at isolating pure chromatin-bound DNA without any contamination of RNA attached to it, which was challenging for me. Yeah.
Okay. I had a question about, you just kind of commented that at least a majority of the transcription factor motifs that you tried did not result in transcription at all in your system.
Yes.
So can you comment on that in terms of,
yeah,
ideally you'd like to be able to answer those sort of more global questions.
Yeah, so ideally we need to look more at, we now have a panel of 300 motifs, but it really looks like that there's a small group of transcription factors that we like to call the big boys, SP1 or GC box, NRF1, YY1, GFX, GFY, CRAP, who are these strong activators, and TADA box. If you have them, you have transcription, boom. And then you have a couple of ones that are sort of questionable, like ETS motifs or BSIPs. Quite often you have transcription, it's a little bit weaker, but sometimes you don't. And then you have a lot of transcription factors where you don't have transcription. And I think if you want me to extrapolate one more step, I think gene regulation has two major things. It's the chromatin, which sort of closes up the DNA and says transcription factors, no, don't play here, and open chromatin. And I think what that shows us is that regulating chromatin opening is actually very important, that a number of transcription factors that have these motifs are not involved in necessarily recruiting the polymerase, but more in opening of chromatin. And that it's actually a small number of really strong activators. Yeah.
They can directly recruit this. Yeah.
Yeah.
Okay. Okay. But no plans for sort of combination type strategies.
There are.
Yeah.
Yes.
Okay.
They are. Yeah. That's of course going to be the real fun thing, is combining them. But doing it computationally, I think the first thing I need to get over is that actually the big boys among each other blend out all the signal. So these 7 loving each other, if you have 2 of them, they're so strong. That's what I really like. So to do so to the combinational, I need to go and analyze more cell type specific gene regulation rather than across the whole cell line. Yeah.
Okay. Okay, cool. All right. Okay, so let's move on. Thank you so much. Okay, so next up we have Owen Chapman. So Owen Chapman is a graduate student with Dr. Lucas Chavez and Jill Mesirov. He's been working to define proteomic and epigenomic features of pediatric brain tumors using tissue from cohorts that have been obtained collaboratively from patients with medulloblastoma and ependymoma. In this work, he's observed extrachromosomal circular DNA, so-called ecDNA in medulloblastoma. And his work focuses on computational methods to detect and measure ecDNA in patient genomic data, and to further define the oncogenic role of ecDNA in medulloblastoma. So great to hear from you.
Thanks for that kind introduction. Let me just share my screen here. Okay, can everyone see what I'm seeing?
Yep, looks good.
That's fantastic. So, hi everyone. Thanks for tuning in and thanks for the kind introduction. I will indeed be telling you about extrachromosomal circular DNA, or ecDNA, which is emerging in the past couple of years as an important cancer driver in a subset of what turn out to be really highly aggressive tumors. So I always like to begin with this image from our colleagues at Paul Michel's lab, formerly here at UCSD and now at Stanford. And what they've done is electron microscopy of a glioblastoma cell line in the metaphase spread. And we can see that in addition to the chromosomes, I really like this picture, you can see hundreds of small circular DNA bodies outside the chromosomes. These are ecDNA molecules. And I'll begin with a few biological properties of these bodies. So the first thing is that ecDNAs dramatically upregulate transcription of amplified genes. Highlighted on the left is copy number on the bottom and transcription on the top of genes amplified on the ecDNA belonging to a glioblastoma cell line. And we can see that EGFR, a known oncogene of glioblastoma, is amplified about 100-fold in the copy number and gene expression more than 200-fold. The cause of this oncogene amplification is likely a property of the ecDNA sequence itself. When a normal cell divides in the center, the mitotic machinery ensures that each daughter cell receives exactly identical genome copies. But because ecDNAs lack a centromere by definition, they assort randomly rather than equally. And so if higher copy number of a given gene on the ecDNA, if it confers a proliferative advantage, then the tumor cell population will accumulate ecDNA copies per cell, driving this massive copy number amplification we see here. So now knowing that ecDNA is oncogenic, quite recently last year, Kim and colleagues screened the PCOG database of whole genomes of mostly adult tumors using a tool I'll later introduce. They find that ecDNA is a frequent and specific feature of cancer cells. This left bar chart shows ecDNA by cancer type in red, with none detectable in normal tissue, but found in up to 60% of glioblastomas. And they also find that ecDNA is associated with worse 5-year survival, controlling for a number of variables, including cancer type, compared to both to cancers without linear amplification and those with linear but not circular amplification. So it's becoming increasingly clear that circular chromatin has unique epigenetic properties as well. Our same colleagues who published those excellent electron microscopy images also performed a TAC-seq and a TAC-C, a variant of a TAC-seq, which inserts fluorescent adapter sequences. Compared to the linear chromatin, a TAC-seq of ecDNA has shorter overall fragments, more peaks, and a stronger overall signal, indicative of relatively accessible chromatin structure. And so now we know that ecDNA likely drives oncogenic expression, both through high copy number and through epigenetic mechanisms. A year ago, I noticed striking enrichment of a TAC-seq signal at key oncogene loci in medulloblastoma, the most common pediatric brain tumor. And so pictured here is one of my original observations at the MYC locus of a set of group 3 medulloblastomas, which are frequently MYC activated and have exceptionally poor prognosis. And you'll notice that the TAC-seq has this striking enrichment of signal around the MYC locus. We also saw a similar pattern at the MYC-N locus in some other medulloblastomas. And so using algorithmic and imaging approaches, we later confirmed these amplifications are ecDNA. And we asked, How frequently is ecDNA present in medulloblastoma? And does it identify patients with the worst prognoses for which current therapies are ineffective? A brief overview of medulloblastoma. There are 4 broad molecular subgroups of medullo, each with distinct patient populations, progenitor cells, and frequent genetic mutations. And in particular, we were interested in asking whether the focal amplifications observed in some medulloblastomas are driven by ecDNA. And we noted also that enhancer hijacking has been noticed in some tumors. And we reasoned that the structural rearrangement that forms ecDNA may enable new oncogenic enhancer promoter interactions. So here's our overview of the study we put together. To look at ecDNA and medullo, we assembled a large retrospective cohort of medulloblastoma patients and model systems from mostly public sources. We identified ecDNA algorithmically by reconstructing circular amplifications from whole genome sequencing data. And in a subset of samples with available tissue, we were able to confirm the presence of ecDNA using light microscopy and obtain epigenetic regulatory profiles of these tumors using other sequencing modalities. For this talk, I'm going to focus on the 2 questions listed here. Which patients do we observe ecDNA, and how does sequence rearrangement affect regulation? Cancer genomics is moving to the cloud, and this has enabled us to analyze genomic sequencing for a large cohort. We started with Amplicon Architect, a tool from Bafna Lab at UCSD for detecting and reconstructing ecDNA. We also use a fingerprinting tool which identifies patients by their alleles at variable SNPs from the 1000 Genomes Project. Both of these tools are wrapped in Docker containers, a technology for the easy deployment of software and its dependencies. And we then deployed these apps to 2 cloud platforms of large pediatric cancer genomics resources. And this allowed us to analyze whole genomes remotely and then download results for downstream analysis. Across our cohort of 468 medulloblastomas, we find that ecDNA is very strongly predictive, both of overall survival and tumor progression. This is true even if we control for age, sex, and molecular subgroup using a Cox proportional hazards model. We find ecDNA in almost 1/5 of medulloblastoma patients. By subgroup, we find no ecDNA in Wnt patients who notably have the best prognoses of the 4. Sonic hedgehog, group 3 and group 4 subgroups had incidences of 27, 18, and 14% respectively. Below, I've plotted a subset of recurrently ecDNA amplified genes in our cohort. Many of these are known medulloblastoma oncogenes, including the MYC family transcription factors, GLI2, TERT telomerase, and CDK cell cycle regulator. We also find a few cellular pathways with multiple members recurrently amplified, including DNA repair genes and p53 inhibitors. We also profiled 31 patient-derived models, 4 cell lines and 27 PDX for ecDNA. ecDNA was much more common among patient models than in our patient cohort, driven by much higher rates of ecDNA in the Sonic hedgehog and group 3 models. ecDNA amplified genes differed as well. Notably, every group 3 model with ecDNA harbored circular amplification of MYC. And there are 2 likely explanations for this discrepancy. It may be that these patients are these ecDNAs arose de novo in some PDX models, or else it may be due to some selective bias in the patient tumors that can be established as PDX models. We acquired additional sequencing data for some of the origin human tumors for which these models were derived, and I'm showing that here on the right. We can see that our PDX results appear to be driven by some combination of these 2 hypotheses. That is, sometimes ecDNA arises de novo in a PDX, as indicated here, but more often it's conserved between patient and PDX. Switching gears a little bit, We also performed Hi-C, a sequencing method for detecting spatial chromatin interactions in the nucleus. Enhancer hijacking has been previously reported in medulloblastoma, and we hypothesized that structural rearrangements forming ecDNA could both disrupt normal enhancer promoter activity and form new interactions. When we perform Hi-C on ecDNA positive tumors, we find the interesting but not unexpected result that chromatin interactions are also enriched in Hi-C as well as ATAC-seq, and that's shown here on the left, or here on the right. The dark stripes here on the interaction map show that the ecDNA interacts relatively frequently with the entire chromosome 8, and indeed the entire genome, not shown. Others have proposed that ecDNAs function as mobile enhancers, regulating the transcription of chromosomal genes elsewhere in the genome. Okay, so left, I've reconstructed the interaction map for MB106, a 6-year-old boy with ecDNA positive MYC amplified medulloblastoma. The ecDNA is composed of 2 segments on chromosome 8, one containing the MYC locus, and the other originating about 15 megabases away on the linear genome. Normally, these 2 regions don't interact, as seen in the unrearranged group 3 sample here. However, in MB106, the ecDNA brings together 2 regions into close proximity, facilitating interactions between the MYC locus on one hand and these 2 enhancer loci from the other segment. Adding Rna-Seq and ATAC-seq profiles, we can see that MYC is highly expressed and interacting with known accessible MYC enhancers in cis, but also, again, these 2 accessible loci from the other segment, which is likely an enhancer rewiring event catalyzed by the ecDNA structural rearrangement. Unfortunately, no model system of this tumor was made, making functional verification of these particular interactions difficult. However, MB106 is not an isolated case. Indeed, we're finding that aberrant chromatin interactions seem to be pretty commonplace on ecDNA structures. On the left, we have a possible reconstruction of the ecDNA in the D458 cell line, and our Hi-C maps identify interactions between MYC, again, and promoter and enhancer regions originating from chromosome 14. We're currently running a CRISPR inhibition screen targeting these enhancers to determine whether they contribute to cell growth. On the right is an ecDNA from a pediatric SHH subgroup patient. MDM4 is recurrently found on ecDNAs from various tumor types and is the likely oncogene here. But we also identify aberrant chromatin interactions targeting LHX9 and KCN2, both associated in peer-reviewed literature with CNS tumorigenesis. And finally, I'll highlight a case harboring 3 extrachromosomal amplifications, 2 of which are shown here. RCMB56 is a PDX model, from Rady Tumor Children's Hospital, originating from a relapsed tumor of an 8-year-old boy with Sowny-Kedrock medulloblastoma. The ecDNAs pictured are detected in both the patient tumor and the PDX. There are many chromatin interactions that cross structural breakpoints, but I'll highlight a few targeting the DNTTIP2 and the KMT2E chromatin remodeling genes. Using fluorescence and C2 hybridization, we stained for genes on each of the ecDNAs. In the leftmost column, you can see that fluorescence of the gene locus does not always overlap the DAPI stain shown on the chromosomes, showing that each amplification is extrachromosomal. By co-staining the same cells, we were able to show that these genes do not co-localize and must therefore be distinct ecDNAs. And we're currently generating single-cell sequencing profiles of the patient tumor and its PDX to understand the copy number heterogeneity in all 3 ecDNAs in this tumor. Our single-cell sequencing data have only just come in, but we've already begun dissecting ecDNA heterogeneity within the tumor. Our preliminary results indicate that single-cell ATAC-seq shows the same enrichment for ecDNA as does bulk ATAC-seq. And we're finding that this allows us to detect the ecDNA bodies at single-cell resolution. I'll wrap up here, but I'm happy to take questions. In conclusion, I've shown that ecDNA in medulloblastoma has the following properties. We find it in almost 1 out of 5 patients at 18%. They usually amplify at least one probable oncogene. They distinguish a particular subset of patients with remarkably poor prognosis. And it's not just the oncogene that's important, the co-amplified sequences seem to be playing a role as well. As I mentioned in my talk, we have ongoing work, which I hope to share with you soon, including functional verification of the enhancers active on these ecDNAs, and single-cell sequencing characterizing ecDNA at heterogeneity within the tumor. It takes a lot of good scientists to do good science, and I'm happy to work with these ones. Special thanks to Lucas and Jill, who are my PhD advisors. Much of the imaging work you saw was done by Josh and Iris in the Shell Lab, and especially Wechsler Rea Labs and Bakchi Labs have been very instrumental in doing some of the functional work that we've been doing. Thanks very much, and I'm happy to take questions.
Great, thanks, Owen. That was fantastic. We have time for a couple questions. And so I'm going to start out with G. Lee, who's asking, did you observe correlation between ecDNA and epigenetic modification, i.e. DNA methylation, histone modification, et cetera, in medulloblastoma?
We have ATAC-seq, which is, as we know, correlated with some epigenetic modifications, but we don't have ChIP-seq or epigenetic modifications per se on these tumors. So I guess I can't really speak to that question. I would expect that given that the ATAC-seq shows this incredible enrichment of accessibility on ecDNAs, I would expect there to be enrichment of other epigenetic signals as well.
Mm-hmm. Yeah, and I guess part of the question is essentially what are the properties that are giving it at such a high transcriptional output, and from an epigenetic perspective, I guess. So are you aware of other data suggesting, for example, Pol2 or K4 trimethylation would be very highly enriched at those sites?
It's an interesting question. No, I'm not aware of other data suggesting that. I think
sorry, so just to clarify that, I have a couple of clarification questions. So the linear amplification of MYC versus the extrachromosomal amplification of MYC, in sort of head-to-head comparisons, those are the transcriptional outputs of those two are different.
Yes.
Yeah.
So one thing that others have shown is that in particular, even when you control for you take transcription per copy of linear versus an extrachromosomal amplification, you see more transcription and greater accessibility per copy.
Okay. And then I had a question about what you were observing in terms of, and I think I might have missed this with the circular DNA being, is it clearly that those are alterations that are observed within the circular DNA itself, so that those promoters and enhancers are being effectively, chromosomal pieces are being effectively resorted in that, or is it that the circular DNA is actively interacting with intact chromosomal areas that are providing transcription factor hubs or other sorts of trans interactions?
You mean those sort of dark cross lines?
Yeah, so I guess I was confused about whether both are happening or.
No, the crosses are the ecDNA, which is usually small, hundreds of kilobases to tens of megabases, and whether it's because there's lots of copy number or because of this sort of rolling enhancer hypothesis interacting with the rest of the genome at remarkably high frequency. And Roel Verhoek's group has come out with an interesting paper lately trying to show that indeed these small ecDNAs are mediating enhancer promoter interactions onto the chromosomal promoters.
Is it clear how that's happening? So when you say mediating interactions, I think that part is a little foggy for me.
Yeah, well, so maybe summarizing their paper in a sentence, their hypothesis here is that because these ecDNAs are very small and not tied to much larger chromosomal structures, they can sort of swim about the nucleus relatively freely, and then bind to I guess promoters where they're, I guess it's energetically feasible. And so yeah, they think that you will get clusters of ecDNA that are binding promoters elsewhere on the genome.
Mm-hmm. Okay, we have a question. Have you noticed any mutations related to CTCF sites or methylation levels in that region and or CTCF binding proteins?
We've not, but that's mostly because we haven't looked for it. I think CTCF chip or related experiment would be pretty interesting to do on these sequences, but I can't speak to it.
Okay. Okay, I think that's, oh, there's another comment. Okay. Is ecDNA amplified itself? Is it self-amplifying? I think it's the question.
Yes.
Yeah, okay.
It doesn't always have to be. Sometimes you'll see or circular sequences that aren't amplified 100-fold, but you do see amplification 10 or 100-fold pretty frequently in primary tumors.
Okay. Has anyone done a competition experiment? I guess maybe single cell approaches might be necessary for that, but where you do sort of a competitive experiment to see, it sounds like from your PDXs that there isn't an incredibly rapid change in the number. But I guess have you, yeah, in terms of how frequently they I don't know what I'm trying to say, how frequently they become more in number. Has anyone actually done that sort of competitive experiment, even in cell culture, to identify one copy versus more copies?
There have been related interesting experiments. In the Michel lab in around 2014 did some work showing that ecDNA copy number in bulk changes in response to drug treatment. So they took an ecDNA positive glioblastoma line, treated it with an EGFR inhibitor, and showed that copy number of the EGFR went away really quickly in the cell population overall. And then when you halt treatment, the EGFR comes right back a lot faster than would be expected from normal cell division.
Right. Okay.
But I'm not aware of any experiments of the kind that you described, where you're asking, how competitive is a copy of 10 ecDNAs versus 20, et cetera.
So we have someone asking, is ecDNA, can it get out of the cell? Can you detect it in, for example, cerebral spinal fluid?
Another fantastic question, which I don't know the answer to.
Okay.
I know it exists in cells. It's really an open question as to whether it can get out of the cell.
Okay. Okay, great. I think we are all set at the hour. Thank you both for really exciting talks and for rounding out our series.
Thanks very much.
Okay.
Appreciate it.
Okay.